Okay, last time we were talking about BK models, in which we mentioned in general that the model is something that we are using to describe certain data or information that we have. Okay, when we are talking about kinetic model, then we are talking about some kind of hypotheses that describe uh, the relationship that we have in a quantitative way and concisely. So, as we mentioned earlier, and let's say in this profile or similar profile or any concentration versus time profile, we have different data points or experiment that we did, and we are using the model to mathematically describe the relationship that we have, in which we have concentration versus time. So we are having a relationship, mathematical relationship, that describing the concentration versus time relationship concisely. Okay, and we will have it as equation. Okay, we mentioned also that we have different type of compartments, and we will talk about compartmental models. The majority of this course will talk about the conventional or empirical compartmental model, but we will talk a bit here and there about physiological based pharmacokinetic. We mentioned that the empirical conventional model is simple. It depends on the data that you have. Usually we use it when we can do much of experiment and we can take tissue related information and samples, and usually it's data dependent, because that's why we call it empirical, because it depends on the experiment that we are doing. When we talked about physiological based kinetic, we said that we usually call it the perfusion or the flow model, because it depends on certain kind of physiological related spaces or physiological related tissues or physiological related organs. Okay, and to describe it, it is more complicated, but at the same time more realistic. And we said it's more complicated because it's experimentally difficult to perform. We need to know certain kind of things about the organ itself or the tissue itself to be able to perform the physiological based kinetic. And we mentioned this, or we show this as an example, or just a let's say symbolization of the physiological based pharmacokinetic, in which we need to know a certain information about the organ. We need to know the flow of the blood to that organ or the perfusion. We need to know the input concentration of the drug, and we need to know the output concentration of the drug, in addition to so many things that we will take it into consideration later, or we will talk about it in details later. We stopped at this point in which we were talking about compartmental based kinetic, and from this point until I specify or mention otherwise, we are talking about empirical conventional pharmacokinetic models, but we will call it as just compartmental models. So unless otherwise mentioned, when we say it's a compartmental model, by default I'm talking about conventional sample model. And again, we mentioned that it's sample because it's data dependent. In usually we don't have that much of samples that we can have from tissues or organs, and we take certain biological fluid and we do its relationship with time, and we will see this in a minute. The concentration of the drug after a given dose, it's defined by certain parameters. As we mentioned before, the parameters are what is constant, that is estimated from the relationship that we have from the variables. If we are talking about concentration versus time relationship, Whatever this relationship is, we can, from this relationship, estimate certain parameters that we usually define the model using them. One of the main parameters that we have is the apparent volume distribution and the elimination rate constant, and we will talk about this later, okay? But just know for now that we usually do some estimation using the model to determine certain parameters to define the relationship that we have. And usually those parameters are considered constant per model, and we will talk about this later. We present or represent our model as a compartment, which is as a box here, and the kinetic proceeds is arrows. So if we are talking about absorption, let's say, which is a kinetic process, we will have it as kinetic process that is shown as arrow here. And if we are talking about the body, let's say, we are presenting it as a compartment or a box, okay? And let's say this other kinetic process is related to elimination. And we will talk about the details later on, okay? But we are presenting the compartment or group of tissues or group of organs in a specified space that are behaving kinetically in the same way, and we are having all of them in one space or compartment or box. And the kinetic processes, we consider it or represent it as arrows that is entering or getting out from that compartment. We may have one compartment, we may have two compartments, we may have three compartments or multiple compartments, but usually after the three compartment model, we don't go into further compartments or number of compartments, because as the compartment number is increasing, the complexity of the model is increasing, and the difficulty of the modeling will be more. So, but we can have as much as compartment as we need in all compartment that we have. Each one of them will present not only a specific tissue or organ as in here in the physiological model, but it will represent a group of tissues in fluids. And when we are saying similar here, we are talking about a similar in a kinetic point of view. They are behaving kinetically in the same way, okay? In a similar way, or like each other. And here we have an example of a compartmental model. Again, it's a conventional model, and it's a two compartmental model. We have one compartment and we have two compartments, and we have in between, we have the reversible process of the distribution between the major compartment that contains the blood, usually, with the second compartment that contains other organs. We usually call it as compartment 1, compartment 2, or central compartment, peripheral compartment, and we can have different, as we said, a group of organs here. With a group of organs here, they are behaving the same. Usually what we have in the central compartment is the highly perfused organs, okay? Highly perfused organs, which means that they have so much blood input in them, so the distribution to them and distribution equilibrium is quick and fast to those organs. Examples, the systemic circulation, heart, liver, lungs, and kidneys. So they are highly perfused organs. In the peripheral compartment, we usually have slowly perfused organs in which the blood takes time to distribute to those organs. So accordingly, the drug takes longer, or it needs more time for the drug to distribute, for the duration of the distribution is more to those organs. So the equilibrium of distribution to those organs is longer. Okay, examples on those slowly perfused organs: fat tissue, muscles, and cerebrospinal fluid. Okay, and again, all of the distribution here, they're of the organs, or whatever that we put here, whatever that we add here, depends mainly on the distribution of the drug, okay? Of course, the blood is perfused highly to certain organs and slowly to other organs, but it depends on the distribution of the drug, which depends in the first place on the physicochemical properties of the drug. For example, if we are talking about highly hydrophilic material, we know that the blood will be here and the cerebrospinal fluid will be here, because if it's highly hydrophilic, if the drug is highly hydrophilic, it will take time to reach the cerebrospinal fluid because it's highly lipophilic as a membrane. But if we are talking about highly lipophilic material or highly lipophilic molecule, we will have the blood-brain barrier or the brain in here. Why? Because the drug is highly lipophilic, so it's reaching the brain or the blood-brain barrier fast compared to other organs. So it depends on the distribution of the drug. So if I asked you if we have one compartment versus two compartments versus multiple compartments, the division and the number of compartments depends mainly on the distribution of the drug as a kinetic process, which depends in the first place on the drug itself. So the number of compartments in the model depends on the drug that we are studying. It's either 1 or 2 or 3, depends on where the drug is going and when it's going to those kind of organs, okay? We mentioned before one major assumption in the kinetic processes, which is the kinetic homogeneity, if you remember, which is in one of your slides here. And we mentioned that this is one of the major assumptions that we usually have, that it's not valid all the time, but it's a general rule of thumb that the equilibrium, the concentration of the drug measured in the biological fluid, which is most commonly in the blood, under equilibrium with the drug at the site of action, right? Under equilibrium, okay? And this is important. It's not equal, it's under equilibrium, which means that the concentration of the drug measured at the biological fluid is reflecting whatever is happening here. Or we can consider or know or assume whatever is happening at the site of action by knowing whatever is happening here in the biological fluid. And we mentioned that this is one of the major assumptions, and it's usually true unless we have some exceptions that we will talk about later. It's an important assumption because usually we don't have the feasibility to do or measure the concentration of the drug at the site of action. This is one of the major assumptions that we already mentioned earlier, okay? One of the other assumptions that we usually consider that each compartment, which is a box, we just mentioned it's our box or it's our compartment, we usually assume that each compartment have a well-stirred definition, or we call it a well-stirred compartment, which means that once the drug is entering that compartment, whatever that compartment representing, either the whole blood or group of tissues, central or peripheral, that once the drug is in, we have instant distribution, instant homogeneous distribution. Well-stirred, it means from stirring that it's stirred very well. So once the drug is there, we have instant distribution there. And there is another assumption that we will talk about it later. And when we are talking about elimination, we usually consider it a first order kinetic or first order elimination. And we will talk about it later, which means that the elimination rate constant is constant. It's not changing with time. And we will talk, we will just leave this point when we talk about the order of reactions kinetic wise, but just add on the assumptions that we usually have, that we are talking about also well-stirred compartment. So when we are talking about the model parameters that I just mentioned in a minute, that such as volume, apparent volume distribution, or elimination rate constant, they are, as we mentioned, estimated from the experiments that we have, such as concentration versus time, okay? Once the parameter once the complexity of the model increased, such as compartments that we have increased, it means that the data needed to be collected more, the experiment that is needed to be done is more, and the parameters that we need to define the model are more. Okay, so again, if the compartments and the model is increasing as numbers, it means the model is more complex. Okay, and if the model is more complex, it means that we need to do more experiments. We need to have more samples and data points. And if we have more samples and data points, it means that we have more parameters to define that model. Okay, and this is one example on that. If we have, this is a one compartment, right, and we have one process, kinetic process, which is elimination process. Oh, okay, so we have one compartment and we only have elimination. One example on this compartment is the, let's use other than blue. Okay, example on that, it's the IV. So we are considering IV bolus because we don't have absorption, right? So an IV bolus model, which is the simplest but the most basic model in compartmental model, we have one compartment here, which means that all the organs, everything in the body is in the same compartment, and we have one kinetic process. If we consider, again, that the volume of distribution and the constant of each kinetic process, whatever this process is, are the parameters that we need to define the model. It means that in this model, we have the elimination rate constant and volume distribution of this compartment. So we have two parameters to define the model. Again, under the assumption that we are talking about volume distribution of each compartment and rate constant of each kinetic process. Okay, in this model, we have the same one compartment model, but the difference that we have absorption process and absorption rate constant, and we have elimination rate constant. So example on that, if we are talking about oral route of administration, right? If we are talking about oral route of administration, it means that we have absorption and at the same time we have elimination, and we just said that we have one compartment here. If we, under the assumption that we have volume distribution of each compartment and rate constant for each kinetic process to define the model, then this will be volume distribution of this compartment 1, we have absorption rate constant 2, and we have elimination rate constant 3. Okay, so again, we said that if the model is more complex, the complexity of this model increased because we added one kinetic process. So again, the complexity of the model could increase or decrease depending on the number of kinetic processes that we have, right? So that's why we have 2 parameters that is needed to define the model compared to 3 parameters that we need to define the model. And here, which is different than in this one, that we are having 2 compartments. So we have 1 compartment and 2 compartments, and the same here, we have 1 compartment and 2 compartments, which means that we have central compartment, peripheral compartment, central compartment, peripheral compartment. And this is a model, and this is another model, by the way. Those are examples of the models just to explain the idea out of it, okay? So this would be why we have 2 compartments versus 1 compartment here. Again, it will be different drug that is going fast to certain organs. That's why we adding them in 1 compartment, and going slowly to other organs. That's why we adding it to another compartment, the peripheral compartment. This would be, again, an example for IV bolus, and this would be an example for oral because we have an absorption process. If we, again, assuming this, that we are talking about parameters to define the model as volume distribution of each compartment and constant, rate constant for each process, kinetic process that we are talking about, then we will have here we will have volume distribution for the first compartment, volume distribution of the second compartment, elimination rate constant, distribution rate constant from the first compartment to the second compartment, so this is 4, and distribution rate constant from the, we have, by the way, we have mislabeling here because this is the direction of the arrow is from first to second, so this is, yeah, it's first to second. No, we are right, guys. So we are right, yes, it's from the second to first to the second compartment, and here distribution from the second to the first compartment. Okay, so we have one model, we have one volume distribution for the first compartment, second volume distribution for the second compartment, elimination rate constant 3, distribution rate constant 4, another distribution constant 5. So just by adding one compartment compared to this model, which is IV bolus but one compartment, we needed 5 different parameters to define the model. And by the way, this is a simple way to look at it. When we will have one compartment versus 2 compartments, you will see what I'm talking about. And here, the only difference between this and that I just added the absorption because we are talking about oral, right? So we will have one volume distribution, second volume distribution, absorption rate constant, which is 3, elimination rate constant, which is 4, distribution rate constant 5, another distribution constant, which is 6. So we will have 6 rate constant. In this slide, the complexity of the model happened because we have extra compartments. So in here, the complexity of the model could be if we compare this to this, or in here, if we compare this to this model, the complexity of the model happened because we increased the kinetic processes. Okay, so increasing the kinetic process, well, whatever it's 1 or 2 or 3, will increase the parameters that we need to define the model. When we compare whatever is happening here in this slide with this one, we know that increasing the number of compartments to increasing the complexity of the model. And if the complexity of the model is increasing, it means also the number of the parameters that we need to define the model are more, and number of parameters more. And if we are talking about more parameters, it means that we need to do more experiments to have more samples. So that's why, vice versa, we need more parameters to define the model. Okay, and we will stop here and we'll be back with the rest of this slides.